pleasant good morning to one and all present here. I, Kanchan Vishwakarma, would like to take this opportunity to welcome you all to the Gyan course, Genomics, Personalized Medicine and Ethics, organized by Department of Biotechnology and Center for Medical Diagnostics and Research, MNNIT, Allahabad. Government of India approved a program titled Global Initiative of Academic Networks in Higher Education aimed at tapping the talent pool of scientists and entrepreneurs internationally to encourage their engagement with institutes of higher education in India so as to augment the country's existing academic resources, accelerate the pace of quality reform and elevate India's scientific and technological capacity to global excellence. Under this program, our course is designed for the participants from various backgrounds. Advances in genomic and clinical science have created an innovative opportunity to tailor healthcare to an individual. Personalized medicine is a strategy based on individual phenotyping of profiles rather than the long-established one-size-fits-all approach. It is based on a multidisciplinary approach, translating advances in basic sciences and drug discovery into development of clinical practice. This course will include basic genetic concepts of gene expression, mutation, and how genes are passed on to the next generation alongside the principles of risk determination. The course will also include the insights into the future perspectives of personalized medicine, pros and cons of clinical genetic testing in prenatal, pediatric, and adult settings, and ethical and regulatory aspects of genomics-based medicine. Now, I wish to extend my warm welcome to the foreign faculty, Dr. Vasu Punj from University of Southern California, USA, and request Dr. Suresh Sharma to greet Sir with bouquet of flowers. Thank you, sir. I would like to welcome our local coordinator of GYAN, Dr. G.P. Sahu from School of Management Studies, MNNIT, Allahabad, and request Dr. Abba Kumar Rai to greet Sir with bouquet of flowers. It is said that all auspicious occasions should commence with the lighting of lamp. So I request all the dignitaries to please come forward and light the lamp as a symbol of imparting knowledge.
शंकरा प्रभृतिभिः देवैः सदा
host faculty and principal coordinator of this course, Dr. Shivesh Sharma, is presently Associate Professor and Head, Department of Biotechnology and Coordinator CMDR at MNIT Allahabad. His research interests include microbiology, environmental biotechnology, development of effective and safe bioformulations, and doses leading to infectious disease management, which consists of identifying the microbial cause of an infection, initiating if necessary antimicrobial therapy against microbes, and controlling host reactions to infection. I would like to invite Dr. Shivesh Sharma for his welcome address.
Esteemed foreign faculty of this course, Dr. Basu Kunsar, Dr. G.P. Saab, Dr. Amber Kumar Rai, Dr. N.K. Singh, Dr. Rugesh Kumar Tripathi, other faculty members of Biotechnology Department and staff members of CMDR, dignitaries and dear participants. Good morning to all of you. It gives me great pleasure to welcome honorable foreign faculty of this course as well as my teacher, Dr. Basu Kunj from Craig School of Medicine, University of Southern California, Dr. G.P. Saab, local coordinator of Gyan program as well as senior faculty member of School of Management Studies, senior member from Department of Biotechnology, Dr. N.K. Singh, Dr. Amber Kumar Rai, coordinator of this course as well as a member of Biotechnology Department, Dr. Rugesh Kumar Tripathi, scientific officer CMDR, other distinguished guests, and dear participants to this 2 weeks course under Gyan scheme entitled Genomics, Personalized Medicine and Ethics, being organized jointly by Center for Medical Diagnostic and Research as well as Department of Biotechnology at MNIT Allahabad. The aim of this course has already been explained by Kanchan, but actually it is to introduce the concept of personalized medicine. In addition, the course will also highlight the ethics and regulatory issues involved in application of genomics-based strategies to personalized medicines as well as its impact on healthcare. I am very happy to share with you that we have received a very good response for this particular course, and in all 106 participants have registered on the Gyan portal, and we have finally shortlisted 65 of them for this particular course. Now the majority of the participants for this particular course are PhD students or the doctoral, they have already completed their doctoral degree, and they belong to University of Allahabad, National Botanical Research, University of Allahabad, National Botanical Research Institute, Lucknow, Banaras Hindu University, Varanasi, faculty members from University of Lucknow, MNIT Allahabad, SHIATS, MMU Maulana, and also from pharmaceutical company, biomedical company, that is Innova Foresight Health and Biomedical Systems, Bangalore. Now as course coordinator, it's my sincere request to all of you to make full use of the time as well as the facilities which are available at the center, interact with the foreign faculty as well as with other faculty members of Department of Biotechnology as well as other faculty members in the institute, and to know various avenues in research, latest developments and future directions in addition to the course contents which will be taught during this 2 weeks program. And I hope that all of you will make full use of the opportunity which is available to update your knowledge as well as skill. And I once again welcome you all to this 2 weeks course, and I sincerely believe that this experience of yours will be a fulfilling and promising. Thank you and welcome all.
Thank you, sir. Local coordinator of Gyan, Dr. G.P. Saab, is Associate Professor in School of Management Studies, MNIT Allahabad. His area of interest includes Management Information Systems, e-commerce, e-business, e-government, IS/IT planning, IT benchmarking, and green IT. I would like to invite Dr. G.P. Saab to say a few words.
Thank you, Kanchan. Good morning to all of you. Dignitaries on the dais, Dr. Basu Kunj, Dr. Shivesh Sharma, Dr. N.K. Singh, for the other colleague, dear participant. It is a pleasure to talk about the Gyan program here. I'm the local coordinator for the institute, as Kanchan has already introduced. And she has also given the main objective of the global initiative on academic network that we are doing here. Under global initiative on academic network, the MHRD has already approved 996 number of programs, and still we are expecting almost same number of another 1,000 number of programs for the phase 2. So this program, of course, was approved in phase 1, and then we are organizing this year. Huge amount of money is being spent for of course, the purpose is the quality education. We are able to invite foreign faculty with rich experience, with expertise, and getting ourselves, knowledge sharing is taking place. So that way, global initiative on academic network is doing good work. In our institute, for the first phase, we submitted around 30 number of programs. Out of 30, we got approval of 15 programs, and this is the 12th program we are organizing. So this includes, of course, all the departments in our institute, including Applied Mechanics Department, Electrical Engineering, School of Management Studies, Biotechnology. So altogether, we got the approval of 15 number of programs, and this year again we have submitted 36 number of programs, out of which 8 number of programs have got approval, and other programs are, of course, in the pipeline. So you can expect more and more number of programs in future, not only biotechnology department, but other interdisciplinary related department as well. So therefore, it's information for you to share this information with your institute, your fellow colleagues, friends, that such initiative is going on, and not only in MNNIT but all across the country, similar programs being organized. I got the information from our foreign faculty that HP University, Himachal Pradesh University is also about to start a program on Gyan. So though final approval they have not received, but they are in the process of getting the program organized. So we are doing good in terms of institutions of India as well, that number of programs that is being organized under Gyan initiative. Another initiative that I would like to share, that you can see that this program is being video recorded, and this video recording will be made available on NDL, National Digital Library. So that's another important information for you in case a few of your friends could not participate due to their schedule, etc. This program will be made available on NDL. Entire program will be there, and then further, I'm sure that foreign faculty will permit you to interact with him in future as well. And then, similar to this Gyan initiative, we have MHRD has proposed two more initiatives similar to this one. One is Global Initiative on Research Network. Similar to teaching learning process, we are going to have the GIRN, that is Global Initiative on Research Network. Under this program, the Indian faculty member will be getting an opportunity to go abroad and see the infrastructure, lab facility, and then at the same time request the foreign faculty to come here, and all the expenditure will be borne by the MHRD initiative that is named as Global Initiative on Research Network. I was talking to foreign faculty about the Vajra program of Government of India. This is another good initiative that MHRD has announced. Under this program, we can request foreign faculty to come here in our Indian university institutions, and the foreign faculty will stay here for 1 month to 3 months, and accordingly the remuneration, compensation, all the expenditure will be taken care of under this scheme. So with this, I thank MHRD for generously funding this program. You can imagine how much funding is going on under this program. For one Gyan program, this program we are spending $12,000 plus amount of dollars. Okay, so $12,000 we are receiving from MHRD, and few other sources of funding is also there. That is of course negligible, but part of it. So you can think about $12,000 and then 996 number of programs already approved, and we are expecting another 1,000 number of programs to be approved in this academic year. So with this, I thank MHRD and Thank you very much for sparing time, and I'm sure all the participants will be benefited out of this program. One more I forgot to mention that we are also in the process of finalizing the credit transfer document. So credit transfer is, as you know, that under this program you are not only going to get a participation certificate, but you'll be having 2 examinations, and based on your performance in the examination, we'll be getting a certain mark sheet as well. So transcript will also be generated, with grade B, B, C, F, etc. So this transcript is useful in credit transfer. One university, if you are attending this course, 2 credit course in this, is it 2 credit or 4? 2 credit course in, 2 credit course here. You go somewhere else, and if there are some course work requirement for your PhD, for some PDF work you are doing, there you can seek exemption, and therefore we will be transferring this credit from our university to the university where you would like to get it transferred. So that is another good initiative. UGC is almost finalizing it. With this, thank you very much, and I congratulate both the coordinator for taking this initiative, and all the participants. I'm sure you'll be getting benefited out of it. Thank you very much.
Thank you, sir. Our foreign faculty, Dr. Vasu Kunj, is presently working as Associate Professor in Keck School of Medicine at University of Southern California, Los Angeles, USA. Dr. Kunj obtained his PhD from Punjab Agricultural University. He served at Himachal Pradesh Agricultural University, Khalagpur, before moving to the Overseas Fellowship Program. He has also worked in University of Illinois, Chicago, University of Pittsburgh, Carnegie Mellon University, Pittsburgh, and University of Southern California. He is also the Faculty Director of Biomedicine. His current research is focused on the development of bioinformatics solution to genomics-based personalized medicine strategies for solid tumors and hematological malignancies. In particular, he is interested in studying functional cancer genomics by intersecting DNA methylation, coding and non-coding RNA profiles, DNA sequence variations, as well as chromatin modifiers such as transcription factors and histone markers. The ultimate goal is to translate the genomic data into clinic by identification of diagnostic biomarkers, prognostic and drug response predictions in cancer. In addition, he is also involved in guiding regulatory documentation for genomics-based personalized innovations. His work provides an important link between basic, epidemiological and translational research. He is recipient of NIH-sponsored grants as well as grants from other funding agencies in US. He has published a number of research articles in reputed journals like Scientific Reports Nature, Cell Metabolism, PLOS ONE, Stem Cell Translation Medicine, Cancer Biology and Therapy, Proceedings of National Academy of Sciences USA, Clinical Cancer Research, Molecular Cancer Therapeutics, Oncogene, and many more. Now, I would like to invite Dr. Vasu Kunj sir to say a few words.
Just post.
Hi, good morning to everyone. Thank you for this opportunity, Dr. Saho, Shivesh, and of course MHRD, so that I can come back and share my whatever I have learned in US for the last 20 years, so I can give it back to young students and they can follow their own dreams, whatever they want to do, in genomics or non-genomics clinics, or they want to do biotechnology. So just so as Kanchan also mentioned, I have my education is from India. So I have PhD from India, a master, BSc, and everything is from India. So I just went and get training from the United States, but I will soon be getting another degree from the US, that is the regulatory sciences, and that is from the USC. So that is kind of okay. So so my basic training is in microbiology. So I did my microbiology PhD, and then I remained in microbiology for a few years. Then I changed it to, went into US with Anandamon Chakravarti. I'm sure you must have read his name in the textbooks. He was the first person who got into the bacteria. So we worked on medical microbiology at that time, and we transitioned his lab into oncology lab, cancer research. So he was basically a alginate bioremediation guy, so he changed it to cancer field. So from there we got, I was lucky to have two patents with him, and one of the molecules which we identified in the phase 2 clinical trials in University of Illinois Chicago Hospital and Children's Hospital Los Angeles. And we do have an industry for production of that molecule and to pursue the FDA for that molecule. Then I moved to Carnegie Mellon and University of Illinois Chicago. So all these different transitions, they gave me an opportunity to move from pure microbiology to different areas of science. So basically I got training in statistics, then computer programming. So I am a little bit comfortable in Python, C++, Perl, and R. And now I'm also getting some background in regulatory sciences. So basically it's a combination of few fields, but it gives me an advantage that I can understand like what a biologist is going to say. It's not just a computer programming that you do whatever someone is saying without knowing what the biology is behind it. So it gives me a chance that I can understand the biology, I can do my computational biology, and then come out with some concrete targets. And I got another patent from USC last year. That is, I developed another algorithm for histone modification. So having said that it's a challenge for you. It's like all the education is from India. So if I can do that, you can also do that. So it's nothing like only US educated students or faculty can do that. So anyone can do that. One has to have the right opportunity, right approach, and right set of mind. So I wish you everyone good of luck, and I think like we will have a good session. Thank you.
Thank you so much, sir. Coordinator of this course, Dr. Kumar Rai, is Assistant Professor in Department of Biotechnology, MNNIT Allahabad. He is specialized in medical biotechnology, infectious diseases, parasitology, histocompatibility, and has expertise in immunodiagnostic techniques like flow cytometry. I would like to invite Dr. Kumar Rai to say a few words.
Dear Professor Ashok Punj, Dr. G.P. Sahu, Dr. Suresh Sharma, Dr. N.K. Singh, and Dr. Samir Srivastava, Dr. Durgesh Tripathi, and dear participants. It is my privilege to extend vote of thanks. First, I should mention Dr. Ashok Punj that he has been very supportive to the department. He has always been kind to offer any kind of support. He has been instrumental in organizing the very first conference of Biosangam 2013. So his support, I don't have words to properly, means the right word to frame that support, but he has been very supportive to us. So I'm thankful to him for sparing his valuable time and coming here and sharing his vast knowledge in the field. I'm also thankful to Dr. G.P. Sahu for taking this endeavor forward and offering his support wherever required. The idea of organizing this workshop was conceived by Dr. Suresh Sharma, who keenly took interest into this and tried his best to make it possible. And I'm hopeful that it would be a very good learning exercise for you all. I'm also thankful to Dr. Durgesh Tripathi for putting his best effort in organization of this course. I must mention the name of student Jaspreet Kanchan and the student those who just sung Saraswati Andana. The staff of the OSD office, Adil, Rakesh, Ravi, and the MDR staff, and actually their names must be mentioned. Without their support, it becomes very difficult to organize achievements. I must mention the name of Shweta Gaur, who actually popularized this event in the University of Allahabad. I also extend my thanks to the whole team of Gyan for taking such wonderful initiative. I also congratulate the participants from various institutes for taking interest in this course and believe that it would be a great learning experience for you. So I'm thankful to you all and welcome to this workshop.
Thank you, sir. This is all here for the inaugural session. I hope you will enjoy this course. And all the participants now are requested to proceed for the refreshment in the terrace. And we will begin our first lecture by Dr. Vasupun sir in 15 minutes.
Okay, good morning once again. So I think like you can take these chairs. One, like people were sitting on the back side, but I speak very loud, so there should not be any problem. You can sit anywhere. So we will be discussing a little bit about the genomics in the context of personalized medicine and what are the ethical issues, socio-economic stigma problems are there. So the way I have kind of organized this course is like I will start with very basic molecular biology, and some of you might feel that, oh, I have done it in my high school, you don't need it, but some of you, I will shortly come to that point, is we need to have some basics, like if someone doesn't know what the transcription is, what the translation is, what post-transcription modification is, what post-transcription modification is. So if you don't know that properly, we will not be able to follow whole genome sequencing or whatever we are going to talk. So just bear with me. We will start with a very basic molecular biology today, but you will then enter into deep into the sequencing, and I want to introduce you to some of the sequencer. I will not go into those details, like there are like several sequencing technologies are here, but I will take it from the concept of the biology, what you want, because you are all biologists, you are not like computer engineers or mechanical engineers, but how you select any sequencer based upon what your need is. And then from that, I think I have divided into like different categories. So this is like for today. And then tomorrow we are going to discuss about the gene regulation, like epigenetic modification and regulation by the non-coding RNAs, mostly microRNA and link RNAs. So all these combinations, so we are starting with the basic, then going into the regulation, and then we are going into like into a real genomics world, that how we are looking into the transcriptome. Like everyone is worried about the gene expression first thing. So everyone has heard about the microarray. So that's why I want to give a kind of an overview, like why you want to move away from microarray to sequencing. Why all the world is going towards sequencing. Why they are leaving the microarray, because it is such an elegant and everything is available. You don't need terabytes of data. You don't need a specialized person. You can manage that data right in a pin drive in the microarray. But what is the fun of going into the whole genome or whole exome sequencing? So those kind of things. And then I will jump into the personalized genomic medicine. That's a very broad topic. I have kept it. So here I want to discuss is what is the concept of personalized genomic medicine. So right now, when Larry Wood proposes it in like 1996, and Craig Venter did the human genome sequence single-handedly, and then where we are after 10 years, are we going to achieve this? We are talking about the personalized medicine, precision medicine, they are all the same thing. Are we going to achieve it? Or what is achievable, or what is going to be just far-sighted, just a fancy term that probably we may not be able to reach ever. And then all this we are talking about genomics and other things. Ultimately, at the end of the day, we are talking about the big data. So when we talk about the big data, is the problem, the challenging part is most of these regular computers, whether it is Mac or whether it is Windows, they don't work. You see, down to one file, basically that's it. It cannot occupy like few fast-acting files. So we need high performance computing, like supercomputers. We work on supercomputers. That is mostly is through programming. Like you do it by Linux, is the most kind of a important tool or operating system that is being used. Windows and Mac. Mac is also good because Mac gives you a shell command which is equal to the Python or the Linux. It's the same thing. So it gives you a flavor of like different commanding languages. And I'm going to discuss actually in the next session like Linux, Unix, shell, Python commands. This is very important. We can talk about the genomics, but we don't like if you get a sequencing done and if you don't know what you are going to do, there is nothing you can do about it. So you are again dependent on a person like me. So it's likely that you may not find anyone close by, and if you find the person will be too expensive and you have to just give up that you want to do it. So you may not, I'm not going to say that you will be perfect at your own, but it will just kind of give you an idea that you can initiate a discussion with someone that this is what is required. If you're talking to me, if I say, oh no, this needs a Linux, this needs like a little bit of Python also, so it's very expensive. And so you can have at least dialogue to talk to them. And if you go back and if you use Mac, not Windows, there is an option also, like I will show you practically demo that you can use if you are fond of using, mostly in India, Windows is used, but Mac is the one that is the key component in here. Not Mac, but the Linux basically operating system. Just to tell you, one of the things is Google also use behind the scene Linux system. So that's why they are so successful. There are some inbuilt positive points of using that system. That's why in Windows you get too many viruses and too many compression problems. But in Mac, we have some bugs, but not that much. Okay, so they have, this is a system. I will not go into those details, like why they are being insulated from that part. But these are the inherent problems. You can read it on the Google, you can Google it, you can find it, and you encounter it. I think if you have Windows computer, you might encounter it every day. So I'm not trying to criticize Windows, Linux, Mac. I have nothing to do with anyone. But I'm just giving you practical examples. So this is what I want to mention is like we will share something about this. Now then moving forward, I plan to give you a hands-on training. How I have one challenge here of the internet connectivity, because they use proxy server, which is not picked up by my university. I cannot do the VPN. So, but I am trying to figure out. I have figured out a way, but we will see like when it happens. But if not, then I will show you on Windows computer like how it works. I have some tutorials already framed and ready to go, so it should be able to. R is no problem. R I have already installed in Windows, so thinking that most of you might be using that computer, so it might be a little bit easy to get to that. And then I plan to give you an analysis of human transcriptome, which is again like basically everyone's requirement. So you need to know like how you analyze the transcriptome. So I will try to walk you through that once the data comes from any patient, from the sequencer, what you do. And if you don't have a programming expertise, what are your options? Like, it's not the end of the world, like, okay, you don't know, like these programming languages I'm talking about that you are stuck, you will not be able to do anything. There are certain wayouts, like the GUI-based wayouts. So I will try to walk you through a very simple exercise that how you can get to a point where you can say, okay, this is our end results. Then I will also do the drug discovery and genomics. So basically, how this drug discovery program has been changed all the way with the help of this genomics targets that have been newly identified. So basically, like if you see, this is all personal genomic medicine. You have to understand one thing is, I will tell you in 2 days, Like if there is a change in the genomics of one person, that drug will act differently on a person, and if B is different, if that drug will act differently on that person. So these all are interrelated things. Then I will discuss specifically the race-based medicine and individualized medicine. So I plan to talk here about few practical cases like warfarin Prolaxa, which is given to patients who are who has a stroke. So in which patients that is suitable, in which patient is not suitable. So then I will walk you through. Now we are talking about all these things that we have falsified. We pass you through these tools, and we have these disulfide. A gene is differentiable, B gene is differentiable. But whatever I am doing is how will it transfer back to my data? It means you want to see in the genome. Like you have a human genome, then you want to see, okay, you are saying A gene is, let's say, upregulated in humans as compared to normal. You want to see that, right? You just don't want to trust me that what is going on. So I will show you a practical exercise where you take the raw data, whatever differential analysis you have done, that gene is showing the differential expression. So, and there are tools which probably you can do also. It's not a that important. You use USC genome browser, IGB. There are several, but I mostly use IGB and use USC genome browser. Then I will also try to walk you through the different components of the genome. Let's say one gene is there, so we are only concerned about like coding region, exons and introns. There are several other things. I will give you a little bit of background of enhancers, promoters, like what they do. Then I will try to introduce you the mathematical concept, like what we are talking about, how these regulations happen. So all these things what happen in the case of cancer, like if you control, if this is an oncogene, if it is methylated, then it is silent, so it is in your favor. But if it is tumor suppressor, it is methylated, then it is going to propagate your cancer. So these all things are highly interrelated. Then I will take a special topic of personalized oncology where I plan to discuss one solid tumor. Solid tumor is where you can physically see the tumor like liver cancer or let's say breast cancer, and then liquid cancer, which is more challenging like blood cancer, each problem. And then I will move to assuming that we have enough background of the personalized medicine and genomics to the regulatory part. So let's say the problem is we have been talking all about that we have created this genomics target or genomic biomarker. Then what we want to do is say all companies want to sell it and make money because they have spent billions of dollars. It's not as simple. Like genomics work is still very expensive. So they want to make a lot of money. So the money can be made only if FDA is going to approve it. Now, if FDA does not have a background or like FDA has no set guidelines, then it becomes very challenging. So I will try to introduce the connection between the clinical genomics and the regulatory body. So that's the kind of a bridge which I would like to make. And then I will introduce the concept of the regulatory bioinformatics. This is a new field that is developing and is very popular with the various regulatory agencies in Japan, in Europe, as well as in US with Africa. I will talk about the test, and then I also want to introduce to you about gene and law. So here I am going to discuss myriad genetics case. Every one of you must have read in the newspaper or heard on the TV BRCA1 case. So they have been selling the BRCA1 testing kit for 10 years. They made a lot of money, but then their patent was challenged. So what Supreme Court did is they give a different kind of like a mixture ruling. They say, okay, test is okay, but you cannot patent the gene. So how these things are going to affect like these research? Because at the end of the day, if we are talking about the genomics, personalized medicine, and all these things, it's all going to affect the patient, insurance companies, as well as the researchers. So if they are not going to get their due share back, then basically we are stuck. Then I think I will discuss a little bit about the microbiome, how microbiome can define the individual patients. This is an area which is still developing, but I think it's good to know because of the biotechnology background, botany, and other background, like how the gut microbe of an individual patient can describe that whether that patient is going to be susceptible to a particular drug or resistant to a particular drug, or that will have cirrhosis or will not have cirrhosis. Then finally, the last time, I think I will plan to discuss very briefly about the mutations in the disease as markers. Before we conclude this session, now there are two important things I will change here. So in the original circular, so there are two tests they said. Now the tests, don't take it as a test as like I'm trying to evaluate you people. So I think basically I'm evaluating myself, like how best I can deliver the message to you. But having said that, I think what I want to do is say I have changed the test from Monday to Friday so that you can enjoy yourself on Sunday. You need not to worry about any test or anything like that. So whatever we do, we have like small fill in the blanks or true and false, whatever we discuss, those kind of questions and answers. And then in the we have the second test which will cover like most of the stuff on the second last day, and that will also mostly have, I'm fond of doing the true and false and filling the blank, but I think like that Sahu told me that you need to give some explanatory questions also. It may be mandate from Gyan or something like that. So we can have some discussion, maybe one or two questions of a discussion point of view, but no prata, nothing. Okay, you need to understand that. So for example, this is what I'm going to do is say, let's say there is one question in this test which is like 80% of students got wrong. So I'm going to repeat that question here too, okay, without saying anything. So my idea is like how good job I am doing this, whether when I'm talking to you, when I'm delivering the message, you are getting it, I need to improve. Okay, so that's basically the pattern I have kind of decided. Now I already gave my introduction. If you have any question, you are more than welcome to ask right now or maybe later. But what I want to get from you is, I would like to have a kind of a basic foundation, like what is your expertise, from where you are coming, and like what you want to achieve from this course, so that I can align my discussion accordingly. There's nothing to worry. 99.9% of the people do not have this genomics and all that. They have heard the term, they may not be knowing. There are only very few people around here who can actually do the interpretation of those kind of data. So it's nothing to shy away or say that, oh, if I say this, what others will think. So it's better, this is the opportunity when you clearly come out and say that, so then I bring out the same level. Now if you raise your level, everything will be going on the top, because I will be talking in like, okay, I'm talking to people like, hi, but if you bring it too low, then I will start discussing the basic concept which you already know. So I think like, but we have to go by most of the majority, like what the thing is. So I think like I introduce myself again. So you know my name, and I'm from University of Southern California, and I'm an assistant professor there, and I work on bioinformatics. Then I have a little bit of knowledge from the last like 15 years, genomics knowledge, and I'm very comfortable in handling that part. So from this course, what I think is, I already gave you one message, clear message is that my all education is from India. So what I got training was from US. I got the opportunity to work with very high class labs. For example, Anand Mohan Chakravarti, Tapas Gupta is a very good clinician. He's a melanoma clinician. And then I went to Carnegie University and then University of Pittsburgh, now USC. So USC has given me a very good platform to diversify my idea and my science. So most of my research or my efforts are supported by NIH, and then it's coming from Tech School of Medicine as well as the Cancer Center, Noise Comprehensive Cancer Center. So I have a good support from high performance computing, if you talk about supercomputers. So without all these intermixing of the facilities, there is no way like anyone can achieve anything. So I am lucky to have USC's so much of a support to get things going. So what I would like to do is that I can start from maybe from here. If you just address these 4 questions to me honestly, so that will give me some confidence, like where should I start, what should I skip. I can modify my presentation, my discussions accordingly. That will be something useful, but pretty cool.
I'm Manoj Kumar. Working in healthcare biology in Bangalore. I have a basic knowledge in actually I have an MS biotechnology. Hello. I am Manoj Kumar. I come from Bangalore. I am working in course at biomedical system in Bangalore. Actually, I completed MS biotechnology. Presently, I am working as a junior bioinformatician. I have basic knowledge in biotechnology only. Currently, I am working in the field of oncology. So, I want to improve my knowledge in bioinformatics. That only I can.
My name is Sijesh. I am working in Human. My position is junior bioinformatician. I have some knowledge regarding genomics since I have done bioinformatics BSc as well as MSc. So I have done analysis on transcriptome and NGS data. So my plan is to get some idea regarding the ethics as well as how the mutations in the gene can be clinically relevant. I need to predict how it will be clinically relevant. Thank you, sir.
Myself, Rajesh, from IMS PhD. I am a PhD student. I am working in modern medicine. So especially I am working in the cancer survival. So I am not background in biology. So nowadays the gene role of the specific statistical and big data. So I am going to use how the modeling in statistics in big data and genomics. So it's in my plan.
My name is Shivam. Currently I am pursuing my BTech from University, Ambala, and currently I don't have any basic knowledge of genomics, and I have come to this course just to learn about different fields of biotechnology and find my field for specialization.
My name is Subhan Bajpayee. I am research scholar in Botany Department, University of Nagpur. My question is, what is genomic knowledge?
Hello sir, I am Soumya Trivedi from the Botany Department of Nagpur University. I am a degree scholar. My work is related to cytogenetics and mutation breeding, and I was interested in knowing how we can link mutation breeding to genomics and what are the how genomics can be applied in the field of plants.
I am going to focus mainly on the medical side, a little bit on the plant side.
Okay.
But the concepts remain the same.
Yes.
I will try to introduce in 1 or 2 cases in plants, but it becomes a totally
A little different, yeah.
Good afternoon. I am Kanchan Thakur from University of Allahabad, and I am a research scholar, and I have no idea about genomics, but I have knowledge about the basic idea of genomics.
Good morning, sir. Myself, Shefali Singh. I have basic knowledge regarding genomics at PG level. I would like to know a bit as to how I can correlate genomics with my research topic, that is on mutagenesis. So So I would like to get a knowledge about it at the molecular level, if it could happen. Thank you so much.
Good morning, sir. Myself, Swati Kesarwani. I am also the research scholar from Ministry of Allahabad, and my research work is based on mutagenesis. So I want also to learn the genomics in the field of plant.
Good morning, myself Meenal Garg. I am assistant professor in the Department of Biochemistry in the University of Lucknow, and I am into teaching biotechnology, teaching and research biotechnology. So I have this information, the basic information of the genomics and molecular biology research. Currently my lab is involved in research, the diagnostic and the prognostic significance in human hepatitis cancer. So we are studying that EM4 mechanisms that is doing using current transition mechanisms, and we'll see how it, the diagnostic and the prognostic significance of this in the heterogeneous disease. Thank you.
Good morning, sir. My name is Saroj Pandey from University of Allahabad. My topic of research is also cytogenetics, and I want to enhance my knowledge in molecular cytogenetics in plants.
Good morning, sir. Myself, Biren Singh Yadav, pursuing PhD from MIT Allahabad. Actually, I completed my PhD work on the plant genomics using microarray and all those things. And now I did some works related to computer-aided drug designing, and I wish to know about the personalized medicine, how we can use means how we can use the personalized medicine, and we can use system like the tool to have a potential ligand, and how we can test the efficacy of the ligand and all those things.
Research from University of Allahabad in plant taxonomy, and I have no idea about genomics. I want to know about genomics and related to plant taxonomy.
Good morning, sir. Myself Shailu. I am from University of Allahabad. I am a PhD scholar, and I also have no idea about genomics.
Good morning, sir. My name is Ravi, Department of Chemistry, University of Allahabad. My topic is statistical synthesis of pharmacologically active compounds. What is the examiner, genetic, personalized medicine, measures, and suggestions? Thank you.
Good morning, sir. Myself Saket Jha. I am pursuing DPhil from University of Allahabad Botany Department. My research work is under the developing a new concept of nanobiopeptide. So I'm just going for a new strain for that. If I'm getting a new strain of PGP, that's still in the studio. So how can I go for a bioinformatics? I just have a little knowledge about bioinformatics. So I just want to go with that thing. So I just correlate with my work with the new strain.
My research work is based on human mutagenesis. I'm from Ministry of Allahabad. I have basic knowledge about genomics, and I want to know how I can correlate genomics in my research work. So,
myself Lily Bharwal, from Department of Botany, University of Allahabad, and my work was also based on induced mutagenesis in human science, and I also want to know that how genomics knowledge is relevant to my study, my research.
My name is Yashwant Kumar. I am a guest faculty in the botany department, University of Allahabad, and I am basically working on career botany. But I want to know about genomics and stuff like that.
Good morning, sir. Myself Santosh Kumar, and I am working as a DPhil scholar in the University of Allahabad in botany department. Sir, as one of my system classification, that is angiosperm kinogenic group, which has been throughout the year, I think 4 versions are based on the gene study. They study the genome science of the organism and produce a classification system which is very valuable for us, those who are belonging to the taxonomy area. So I would like to enhance my knowledge about how the genomic study they do and how the research papers the companies got selected to produce a classification system to help us. Thank you, sir.
Good morning, sir. My name is Vijaya Yadav. I am a DPhil scholar from the University of Allahabad. My
how can I implement in my research?
Good morning, sir. Myself, Jacinda Kaur. I'm from University of Colorado. I'm doing research on astronomy of energy storm. I would like to implement this knowledge from the course, relate this with my its knowledge.
Hello sir, I'm Meera Anand from Allahabad University Botany Department, and I'm working on nanoparticle toxicity in plants and their detoxification pathway using stress hormone. I need to know about how I can use genomics in my research.
Myself is Shweta Kaur, and my work is on stress physiology in plants, and I want to know some molecular genomics.
Myself, Sandhya Rai, and I'm doing PhD from MIT College, and I have some basic knowledge about the genomics, and for this course I just want to expand my knowledge and want to use it in this my research.
Myself, my name is Ishwarya Sharma. I'm currently pursuing my MD degree in biology from Allahabad. I would like to explore the area of genomics where I can correlate with my research.
Good morning, sir. My name is Shay Dwivedi, and I have completed my master's in biotechnology from University of Allahabad this year. And I would like my knowledge regarding genomics has been quite limited, but I would like to expand it using this course. And I would also like to know how can I use it for future research, framing future research proposal relating genomics with cancer.
So my name is Paula Mishra. I am from the University of Allahabad, and I am a PhD scholar in the Center of Bioinformatics. I have a small knowledge in the genomics, and we work on the proteomics, and I want to just comparative analysis of the differentially expressed transcriptome and microarray analysis.
So you integrated the genomics with the proteomics, is that right?
Sir, mainly my work is focused on proteomics, but I want to analyze the microarray data and transcriptome analysis.
Yeah, that is something very challenging.
So
But
my
yeah, we
work
have
is,
done it.
okay. So my work is totally on tuberculosis targets and its transcriptome analysis. So I want to just apply what you are teaching on my research.
I am Anamika. I am research scholar at Center of Bioinformatics, University of Allahabad, and I am working in the field of genomics as well as proteomics. And I know very good, even I know I am working in the field of genomics and proteomics, and I am asking you to take your knowledge, your idea, your teaching so that I can implement people by myself.
Myself, Chandavani. I am from University of Allahabad. My research topic is based on immunity. A little bit idea about genomics, but I want to know more about
Good morning, sir. I am Valusana. I am professor in University of Allahabad, Department of Biology. Sir, I am knowledge in genomics and I am basic knowledge in genomics.
Hello sir, myself Vaishali Yadav. I am also from University of Allahabad, Department of Botany. I am working on plant nanoparticle interaction. Here I have come to know how we can correlate the plant interactions with nanoparticle and genomics.
My work is related to accurate data analysis, and my purpose to join this program to learn about the R packet or Linux initial command, and
Myself, I am Nitin Soy, a research scholar at MIT Allahabad. Recently, I was working on phytic acid
colorectal cancer. And I have a basic knowledge of genomics. And I have to guide a lot of things in my work. So I'm here to study and provide my knowledge.
I'm working on leukemia, and right now I'm working on DNA, PCR, and I want to know how to computerize expression level data of genomes, and I want to know how genes are related to pathogenesis or pathobiology of any disease.
You will get a good information from it. If there is a special topic and we have our own work on blood cancer.
Good morning, sir. My name is Neha Upadhyay, and I am pursuing PhD from NIT in the area of environmental microbiology. I have very basic knowledge in genomics.
Good morning, sir. I am Uday Verma, with PhD from NIT Verma. So I am interested to know about the comparative analysis of the organism, how we can use bioinformatics.
Good morning, sir. I am Amit Kumar, student PhD from NIT Verma. My work on gene silencing and reading the function of different isoforms generated by genetic splicing. So this knowledge applies in my
Good morning, sir. My name is Patel. I am from Banaras University. I am pursuing my PhD in human genetics. I am doing my research in human genetics. I am working to explore the genetics behind human gene set. I am also have bioinformatics background at my teaching level. This course will be helpful to make a bridge between these two fields, bioinformatics and human genetics.
Hello, good morning, sir. So I'm Shruti Jain. I'm a PhD scholar in Motilal, Pune, and I have worked on transcriptome data and selected some genes for gene expression. So my work is related to silicon-mediated pesticide toxicity, elevation of pesticide toxicity in plants. So I want to use this other genomic techniques for my work related to transcriptome and other genomics tools.
Good morning, sir. Myself is from Professor Ashton Degree College. I am a day scholar in the Department of Biotechnology, and I have very less information on the genomics and I here to study.
Okay, I think that gives a mixture of, like, there are few people who probably know a little bit about the genomics and they are kind of working on that. So I will kind of, but I think I don't have much in the plants because most of our work is on personalized medicine is of course medical based and it's translational value. So we have never explored the translational value towards the plants. I will see, like, in some of the lectures I try to introduce some concepts in the plant side, but I think like it becomes too deviating from the main concept. So, but we will see like how it goes. So I will kind of keep it a balance. So this lecture may be very basic. So I will kind of speed up. We are already very late, so basically I want to give you kind of an idea like how from basic molecular biology we have moved into this genomics area. A little bit of the historical background, a little bit of like what was done previously. So still some of those old concepts are good, even like we are talking about the NGS and all that stuff. So if you want to define the molecular biology, basically that is study of the macromolecules. It can include your proteins as well as DNA as well as RNA. And what we do in this case is so we try to understand their role in life and like their mechanism of function, so how they interact with each other, and try to develop the cells, divide the cells, or give you the output whatever you are looking for. So it's a very broad field, and it's an microbiology itself is an interaction genetics, biochemistry, microbiology. Microbiology is because microorganisms are the standard organism tool for studying any microbiology concept. So I basically kind of divided this into a little bit of history, then DNA, RNA, very briefly. I'm not going to go into detail of like DNA structure, RNA structure, and DNA. We can spend like whole few months on that. Then how it moves through the genome, and then human genome project, which is very important. And then just one slide of prokaryote to eukaryote, so that's kind of really important in here. And central dogma we all know, and then mutations, and then how we correlate with the functional genomics and transcriptomics as well as proteomics. So if you look at very quickly about historical perspective, so first was the invention of the microscope in 1665. Then cell was discovered. They named that every living thing is made up of a cell. Robert Hooke. And then they also gave, then came the discovery that all the specific tissues are made of identical cells. The cells are, they have certain phenotypic as well as genetic makeup similarity in the particular tissue. Let's say breast tissues, they are like memory tissues, so they are made up of particular tissues. And then Mendel gave the basic rule of heredity. And then in 1869 it was found that DNA is the basically genetic material. They identified it. So then 20 amino acids structure was basically identified. So that is the basically we got building blocks. Then chromosomes are identified as a unit of heredity. Then there was a discovery of RNA. Then genes that can make proteins. So basically, every there's a so later on, like Harvard Corona gave like treatment code that can code it to mRNA and then translate it to the protein. Then binding of AT and CG was discovered. So DNA structure was discovered by Watson and Crick. We all know about that. Then there was discovery of ribosomes. And Baltimore discovered restriction and we basically revolutionized all the molecular biology. So you can have DNA from specific places, put it into something and make something, combine DNA technologies, like kind of revolutionized. Then gene splicing, this is kind of very important, very complicated, and has a very key role in the current scenario, especially when we talk about this personalized genomics. So I will talk a little bit about the detail in this one. So basically, here exons are joined, introns is looped out. That's what called as splicing. So here, like we are now entering towards what we are going to talk in the next few days is automatic sequencing. So Larry Hood was the one who basically first did the automatic sequencing of the DNA. So Larry Hood is the one who proposed the concept of P4, that is precision medicine and personalized medicine. So then in 1986, Human Genome Sequencing Project was initiated by the US Congress, and Craig Venter, so he first sequenced the bacterial genome in 95, and the yeast was in parallel sequenced by another group. And this is the guy, Human Genome Project was completed in 2003. So Craig Venter single-handedly completed the first human genome with $2.9 billion, and it took 10 years. And he is still in the Craig Venter Institute in California. So he has now made another discovery where he has made a synthetic bacteria. So what he did is, he has all the essential DNA segments, put them together, and he has shown that the bacteria can divide in bacteria. So that is going to revolutionize all that, whatever we are talking about, personal medicine, all that stuff. So in parallel, human genome, Drosophila, mouse, and rat genomes were also sequenced. So basically, after 2004 or 2006, this sequencing just take over everything. So everyone is just talking about the sequencing and sequencing. So this is what I was trying to tell you. This is the guy who basically revolutionized all the sequencing. So it took him 10 years, approximately $3 billion on the project, just to sequence his own genome. And later on, Crick also has his own personal genome sequence. So Crick is still alive. Now, we have three major key players in molecular biology. So our life depends on these three critical parameters, DNA, RNA, and protein. So they are highly interrelated in a cell. The cell does not have a brain. So it does not have a brain. So how it decides? So basically what it decides is, it's doing this cell signaling. The DNA gives some signal to RNA, RNA gives to protein, or somehow they are interrelated. There are certain pathways, paracrine, autocrine pathways are interlinked, and then that's what we call as cell signaling. So it decides how to talk from cell to cell, within cell, how to build the cell, which cell has to, if there is something wrong, then the cell goes like on a human progression pathway, or like how when the cell has to get the food, then it has to turn off that signal when it doesn't need any food. So all that is cell signaling mechanism. This is a big effort in most of the labs, in India, abroad, everywhere, cell signaling labs, most of the labs. Someone is working on NF-kappa pathway, someone is working on apoptosis pathway, someone is working on TGF-beta pathway, someone is working on ER pathway. So there is a lot. It's never ending process. So there are a lot of pathways that are affected, and the only challenge here is, like, it's never concrete answer. Let's say you say NF-kappa pathway is affected by A molecule. So it may be like NF-kappa is not a direct target of the A, but it may be a direct target of, secondary target of some other gene or some other molecule. So that becomes very complicated. You can block a pathway. Let's say you block that pathway. So it may be, in addition to that pathway, it blocks some other pathway. So basically, it becomes very complicated, and that's how we are mostly now interested in kind of the genomics, that we can design any treatments directly towards an individual patient rather than just a broad base for all the population. This is the central dogma. Most of us know that DNA to RNA transcription, then protein, and proteins mostly play the function, whatever function they have to do. Now, very briefly, but it is important that we should understand, we should refresh our memory. What are the prokaryotes and eukaryotes? Prokaryotes they do not have the nucleus, but eukaryotes has a nucleus. They can be single or multicellular. Then there is one piece of circular DNA. They have chromosomes. This does not this lack post-transcriptional modification. Like what will happen is, as we have seen the central dogma, DNA to RNA. So they don't have any post-transcriptional modification. Whatever has happened has happened. So it's not going to change that product. But here, gene splicing occurs. So that's where the organ splicing and eukaryotes, humans happening, and that's more challenging to deal with in our personalized medicine effort. Because if you try to target one gene, if it has a multiple splicing form, then you have to target all those genes. Okay, so it's kind of like very, very difficult. Now here we have bacteria and virus. Here we have plants and animals. There is one category which probably some people take it as it can be prokaryote. Some people consider it as a different category. That is the prions. So prions are nothing but just they are the replicating protein particles. That is what their genetic material is. That is what everything is. So it's still not clear how they propagate, how they multiply, what to talk about their like genomics level or proteomics level efforts are. So still there are very select few groups that are working on prions. One is in California and another one is in Germany. So there are few groups that are working on that. So it's kind of very tough to work in that prions, but they produce very deadly disease in the humans. Now there are few terms. I gave you a hint about one term, which is the post-transcriptional modification. So what happened in the eukaryotic cells, DNA to RNA to protein? Central dogma, we have to have our own tips. So everything is going to revolve around that part. So when we are saying the post-transcription modification, that means once the DNA is converted to RNA, there is some change in the mature RNA that can happen. So there is a, prior to the formation of the protein, there are some changes that can be introduced. Now you have to understand the challenge here. If you target the transcription factor, but there is a post-transcription modification, so your efforts are not going to work. Okay? So similarly, post-translation modification. So this I will discuss in terms of the histone modification. So what happens is a protein is made after post-translation. There is a change in the modification of the protein. So there is a one histone like H3, K24, ME3. There are different kind of histone, but I will discuss with one example. We have also published in Genes and Development paper. So I will tell you like how this post-translation modification has changed the genome-wide scenario. So you have to understand this part also. If there is a post-translation modification, how you are going to get it at the genome level. So it's there. So we are like complicating 3 or 4 different phenomena and trying to see that histone modification is occurring. But we have shown that that post-translation modification in that particular histone specifically enhance affect the genes by depletion of the histone in cancer-related genes, either in the coding region or in the promoter region, or coding and promoter region both. So if it is in the coding and the promoter region both, then basically we don't have a phenotype what we are expecting. But in the promoter region and the coding region separately, we have the phenotype that is expected. That means we did it in terms of the osteoclastogenesis in the bone form, because what will happen is say some of these proteins they are metastatic, so we are trying to address the metastatic in terms of the osteoclastogenesis. I will not make things too complicated, but I will show you a picture and then I will come back to the post-translation modification. So this is important. It's very basic. So what is going on here is, so this is a very simplified structure of a gene. Okay, I'm not talking about anything UTR or any enhancer or anything at this stage. So first of all, we need to get is, so what happened in the eukaryotic cell is, so these exons, introns, they will be looped out, and only exons are there. So this is what we call it as splicing. Okay. Now there will be another concept. Now it can happen is only one intron is spliced, or this can be separate. This can be separate from the same gene. So it can be like 2 are combined, 1 is combined, or both are not combined. So it can, there can be like several instances that can happen. And mostly genes have several exons, unless you are lucky working with a gene which has only one exon. Like, but there are very few genes which are. There are some genes, but they are not too many genes which are like having a 1 or 2 exons. There are other multiple exons or very common. So all these. We have talked about prokaryote, eukaryote, we know the molecular biology, and we know like now Craig Wendler has sequenced the human genome. So will the efforts end there? No. There are different people, different research groups, so they try to generate the data depending upon their research interests. Okay? So what we have is, so this is what we call the genome database. So we are slowly going into that publicly available databases. So you have all these databases available like eukaryotes under NCBI, bacteria, archaea, virus, viral, plasmid, and of course human is there. Okay, so human is separate. Now, if you try to work on genome databases, now you will be thinking that okay, if I want to work on human is a big genome, so I will not have too many resources. Or mouse genome or rat genome, but bacteria will be very easy. Okay? It is the case in the reverse. What is happening is for human, mouse, and rat, especially human, everyone is interested, and we have very well defined tools, very well defined resources, UCSC genome browser. There are several other databases. TCGA is there, ENCODE is there. So they mostly deal with human, mouse, and rat also. So they are available. But if you come to the bacteria, it becomes all customized. Bacteria and virus. You cannot find like resources sitting there. Now the way these databases are, so they have the patches of the contigs sitting there. So you have to do a lot of customization at your own. But fortunately, these genomes are very small. Viral genome is very small, bacterial genome is very small. So you can easily handle that. But that is something no one would like to do that, waste the time on that. Because that is, if you reconstruct it, you redo it, you cannot publish that because it's already there. And it's just for your tool that you have generated. So anytime I work on these things, I hate to say that it is more work than working on a human genome, rat genome, or mouse genome. So it's the other way around. So now there are two important molecules that we keep on repeating. That is DNA and RNA. Now, if you look at the gene, so what is gene? Gene is a functional unit of heredity. So basically, that is one which regulates, which has all the signaling information to code for which proteins to be coded, which genes has to be turned on, which genes has to be turned off, which genes are good, which genes are bad. Then chromosomes basically encaps this DNA. So it has the DNA basis. So it basically has all the instructions coded in. Now, if you look at the nucleus, I want to touch base on this. Like what will happen is DNA is very long. Okay, so if you isolate a cell and you unloop the DNA, it will be several miles long. So all this DNA is tightly packed on the chromosomes in the nucleus with the histone proteins. That's why I have mentioned the histone proteins. So if anything goes wrong with the histone proteins, that's going to affect your genes. Okay? So that's why, like I'm saying, is all of us know about the gene expression. Okay? Microarray or sequencing, we have tumor, we have normal, one gene is up, another gene is down, we are very happy in that. But in reality, it is not that simple. So what will happen is we are not taking into consideration of when we apply any drug, like if it is the histone targeting, let's say histone methylase is there. So that is going to affect not only few genes, it's going to affect the reorganization of all the molecules. So then basically you have to control it. Now if you're targeting only the expression, basically we can say that, oh, we find out 10 genes of tumor versus normal, we can target those genes and this is how mostly the thesis ends up. Like you say, oh, we have find out 10 genes, we have revolutionized the world and those things. But in reality, with those 10 genes are just the signature markers of that confined cohort study. Okay? So it's not going to be, it may not be the scenario that you just address those genes and then it will solve all the problems. So this is another inherent problem in case of the personalized genomics. So one thing is biomarkers have made very good progress in terms of personalized genomics. But as far as the therapeutic purposes is concerned, they are still not at a very translational level because of these genomics complexities. So it's always an ongoing process. Now genotype is basically the genetic makeup of an organism. Then phenotype is like how any genomics or any genetic constituents, they express the phenotype. We can say that as we see when once we are looking at apoptosis, whether it is apoptosis or whether it is some other kind of cell death, or what is cell may enlarge when it's going from G1 to G2 phase. All those phases, they have different phenotype, or if you treat with the drug, they can shrink. So all those things that happen in the phenotype, which is kind of very useful for us without going into too much of details. You can find out, okay, your cell has died, your cell has proliferated, or your cell has shrinkage, in between that, if you increase the dose, decrease the dose, those things. But if there's no phenotypic change, then it becomes more challenging, and then you have to look at most of the genotypes, like what you have to do, go back and do stuff with that. So this is the basic structure of a DNA. So we know that it is ABCG, and A combined with D and G combined with C. Watson Crick gave the structure. Now something about the RNA. So RNA is the only molecule that has both catalytic activity as well as it can act as a information storage like DNA is. So there is some concept going on. Some people, some investigators, some research groups, they think that it is not the DNA, it is the RNA which is the building block of the evolutionary trend. So with time it has kept on changing, and that's how we have all developed. But certainly, like most of the people believe that it is the DNA. So, but I just want to give you an idea that concept is there, and this is the functionality of the RNA. That's why we mostly go after the gene expression. Like everyone has heard about the gene expression. So another important feature about the RNA is it is a single-stranded molecule which basically goes back. So it will give you a looping structure which we call as stem loops. So this is the only molecule that can give this kind of structure. This is what is known as a Scamby structure, and it is used for its own stability and function. In RNA, we have in DNA we have ATCG, but here uracil is replaced with T from T. So we have ACG, and then it combines with GC and A. Okay, so this is an important change from DNA. There can be there are several types of RNAs. I'm not going into details of all of those, but I will give you a little bit of idea when we discuss the non-coding RNAs, that there are like several types of the RNA. But there are three important RNAs that we need to understand what they are doing. One is the messenger RNA. So basically it takes a message from DNA, deliver it to the ribosome. That is what we call it as a messenger RNA. So basically it has the information of the genes, what they are coding for. They are coding for A protein, so they have that information, they deliver the message. Transfer RNA, they are the intermediary RNAs. That's why they are known as kind of a middleman. So they have a particular amino acid and its corresponding mRNA codon. They carry an amino acid on one end and deliver it to the ribosome. So basically they just carry the information and deliver it. Don't do anything. Then ribosome, initially ribosomes ribosomes were considered to be at one point they consist of proteins and a little bit of the RNA, but now mostly predominantly it has been found out they consist of RNA. They are consisting of RNA. So that RNA we call it the ribosomal RNA. So that's why we are saying that it can do both catalytic as well as the information storage. That's why mostly RNA plays a key role in drug modulation. Mutations play in part of the DNA a key role in modifying the therapeutic targets or identification of the biomarkers. So we discussed about the nucleic acid. Now all this DNA and RNA is in the cell. Now if you look back, I will move back to DNA and see that cells and DNA. So human body has approximately 100 trillion cells. There are 46 chromosomes, 23 pairs, which will constitute your genome. So they are in the chromosome that contain DNA. The important point here is every cell except a few in individual contains the same exact genome. Okay? But what we see here is like someone is tall, another one is small, someone is fair, someone is too white. Like you can have white population, you have black population, depending upon the race. Or someone is even more intelligent. I'm throwing a very controversial example. Someone is more intelligent, another is less intelligent, or someone food habits are different. So their outlook is different. how these things are decided. So naturally, there are certain cases where the cell has different genetic material than a second person. Okay, so we are going to discuss this part like genetics and genomics, why humans are different, and how you can identify normal from disease from the normal. So that's basically the key part in our discussion. So we have been talking about this genomics. So what this genomics means is, so it is nothing but it is a structural and functional study of the genome. Genome is, if you go back, 46 chromosomes which has a DNA. Okay, so there are few subfields. There are several subfields, but there are three important subfields are structural genomics. So when we say structural genomics means we are studying the structure of the genome. So basically you consider, like as I have mentioned, is not only the DNA, the RNA structure, how the structure is compacted with what is the role of the histones and all that stuff, like which proteins are responsible for their compaction or for their activity when it has to be released. So all that structure remains intact. Functional genomics is like very popular, and most of the research labs try to work on the functional genomics because that gives you transcription value. You want to find out how particularly a group of genes or a genome is going to give you a functional parts, like let's say it's going to give you apoptosis, cell death, or what it is going to give you. So what is the function of those genes? So you can study the function of a single gene by knock in, knock out. So all those methods. And then the third, which is getting a little bit more interesting, is comparative genomics. So that's where like most of our stuff is lying. So we want to compare the disease and the normal to both the genomes and try to find out the difference. We can find out the difference in terms of mutations. We can try to find out the differences in terms of regulatory elements. We can try to find out the differences in the structure, like how it is organized. We can try to find out like how different differences in those ones can be translated back into the phenotype. So that's kind of like a key question. But I will come to another point. There are new mutations that has come up. I will also discuss a little bit about the regular mutation, but there are mutations that new concept is driver mutation and passenger mutation. That's the key part in personalized genomics we have going to. So now when we coin the term genetics, so genetics is nothing. So here we are taking into consideration, let's say, one diseased person or one normal person, but we are not taking into consideration the population. So if we take into population, let's say Asian population, that will become a genetics. Okay, that's what all the GWAS studies are. Genome-wide association. Now if you take let's say white Caucasian or Korean or African Americans, so you take into that population-based studies, or like country-wise Indian population, US population, or any population. So if you take into consideration those, then it becomes the genetic studies, and that's what like someone also mentioned here. That I think a couple of people are working on cytogenetics. So that's basically that you are trying to do it rather in the plants. Those are population-based studies. So you have to understand the complexity of that system is it will have a lot of variation. It will have a lot of clinical covariance. If you go from race to race or like population to population, Asians will have different food habits, different body makeup, different body resistance. Probably like if you go to US population, they may not be resistant. Like that's why you must have heard like whenever someone comes to here, don't consider my statement on the other side. So they normally demand good water is polluted, they get sick because of the pollution, although even if they are from here. So they have a different body adaptivity, their immune response is acting differently. So all those clinical covariates has to be integrated. So that means a lot of statistical expertise to be used, like which clinical covariate can be taken out. So we all talk about the mutations. There are several, a few who are working on mutations. If you work on like few groups of cells, that is fine. But if you take care of like the population, then you have to reconstruct a statistical model by which you can say that which exactly is the true clinical covariate that is responsible for that mutation. So I touched about this part. The important thing is how these two individuals are different. We are saying that 99% of human genes are conserved. Then the second important part is like if A gene is there, let's say CNIC is there, CNIC will do the same function whether it is Caucasian, whether it is Asian, whether it is Korean, whether it is woman or whether it is man. So it will not change. Okay, the same gene will keep on doing the same function and it will not spontaneously develop, like it will become from oncogene to tumor suppressor in some cases. It's not going to happen spontaneously. So there has to be some trigger somewhere which will change some of those genes to come from tumor suppressor to oncogene. That's where the cancer is. So I will keep on adding more and more examples of cancer because there are a lot of studies, a lot of literature, but I do work on cancer as well as other diseases. But cancers are like easily very like elegant example. So that will hold true for most of the other diseases too. So they will not change spontaneously, like CMYC has changed with the evolution and suddenly now it was 10 years back it was tumor suppressor and now it is oncogene. I'm just giving a hypothetical example. That's not going to happen. So we know that all the living things has evolved with a slow evolutionary process. Then what the hell is going on here? Why one person is oncogenic, other person is not oncogenic, in the same DNA, same RNA, same 99% is conserved. So what is going on? So the key lies in, I'm sure all of you must have heard about this thing. So 97%, most of the genome in any individual is identical. There's only a fraction, like approximately 1 to 1.5%, they are different at a single base pair resolution. So when we say single base pair resolution is, if you see here, like this is showing a single base pair resolution. Okay. Now if you start looking at a single base pair resolution in the genome wide, you can find millions of SNPs. That's what we call as SNPs. So the question then arises is, which SNP is going to work for that particular disease? So as I mentioned, like there are 11 to 12 million SNPs so far has been identified, and most of them, not most of them, like I would say majority of them does not have any functional role. Okay, so this is like I would give a statement, but with my due apology, like I'm not against anyone or anything like that. So most of you will see like SNPs, these GWAS studies, you will identify the SNPs, there is a Nature Genetics paper, and you move to the other disease. You want to find another SNP. So what will happen to that SNP? Nothing. And when like people like me, you goes to the literature, you find, oh wow, this is there. Because the functionality is the main part which you can do, but there is a success too. Like for example, AMD, there is a SNP that has been identified. Okay, acquired macular degeneration disease, which is in the old people. So this is an eye effect, there is a fluid that comes out, and the person starts seeing blood, and it can go blind. But that has been identified. Okay, so I'm not saying that everything, but the functionality is very time consuming, very expensive, and no one wants to do that. Okay, that's why we have like, so the easiest part is you identify the SNPs, match it to a particular population. Let's say Caucasian population, Korean population, woman, man, different age, smoking, non-smoking group, and say that okay, this SNP is associated with the development of this disease with this population. That's it. You have a Nature Genetics paper. That's it. Okay, but the important point is functionality. Now, if you have to test this functionality, it needs one RO1 from NIH and an effort of several years. Okay, so that's where like there's a little bit of a disconnect. Although the SNPs are very elegant studies, but there is a disconnect between this type of studies and exactly the translation way. Okay. Again, like I'm not saying that they are not good, they are not good. So there are people, there are very high profile groups that are working on that, they are doing pretty good jobs, but that is kind of like, it's my personal opinion, I think this is a disconnect. Like you can go and Google these SNPs, you will find, oh wow, I found it. But then you start reading, you spend 1 month in reading, going back to literature, and then you will reach at no point. Then you have to find out whether that SNP is nearby a enhancer, promoter, coding region or non-coding region. Now if that SNP is in the coding region, then you are basically in a good shape. Okay? But 99% of the cases it's not the case. It is mostly the regulatory element that is affected by this. And that's why I try to build up a case that I try to bring in the regulatory elements, histone modification, promoters and enhancers. You see like how I have built up a basis, and then try to wrap up everything in a, just like packaging in a nucleus. Okay. So one of the key points is keeping aside that statement, but these SNPs are responsible for differential behavior of any individual. It can be disease, normal. So at least it shows the association. As I'm telling you, let's say in liver cancer patients, A SNP is there in the tumors, B SNP is present in like 10% of the population, but it is present in 90% of the population. So we can say that this kind of SNP has a predisposition for occurring the liver cancer in those patients. Yes? So they are saying that we need to break. I think we are getting late, but we will continue this. So let me just take to a point, and then we can take a break for a few minutes, and then reassemble and continue our whatever we are doing. So the second concept comes in that what will this is a single base pair resolution SNP. Now it can be deletion. It can be any other mutation. It can be base shift transposition, or there are several now structure changes are there. So I'm just giving you a very basic. The mutations can be two types. One is germline, if they are in the germ cells. So those mutations will be carried over from generation to generation. But somatic mutations are in the somatic cells. For example, in the breast or like in any particular tissue. So they don't occur in the germline cells, and they do not propagate from generation to generation. So unless there is some trigger, so that's where the things come in. So somatic mutations are the one we are looking at. Let's say in the cancer we have seen that. So we will say that okay, these are the cancer mutations. So they might have been triggered by either carcinogenic agents, or exposure of UV light, or exposure of some chemicals, or because of some genetic effects, or like genetic predisposition. But those mutations will be present in those cases, somatic mutations. So that's why we have a separate database for somatic mutations. Just quickly, we all know about this. So this is like what will happen in the mutation. So you can have a point mutation, it is a single mutation. Indel, deletion. So everyone is following what I'm trying to say, like indel, everyone knows? Anyone who doesn't know indels? Just be. Okay, so indels, what happens is when we have a stretch of DNA, ATCGC, let's say GCT is deleted. Okay, somehow by any means. Let's not worry about that. That is known as indel. Now indels can be short indels, long indels, whatever is the key part is that particular DNA portion is deleted. So once it is deleted, you have to understand that if it is throwing the protein, if the protein is coding, it can throw the protein out from the coding frame. It may not code for that particular protein, or it can code for a defective protein. So those things can happen. The same is the homozygous relation. So we know that every gene has 2 alleles. So homozygous is like in the both the alleles are affected in the mutations. Hemizygous is only 1 allele is affected. So it can be translocation, like one base pair from the double stranded is translocated to the other portion. So there are other structural variants which I am not going into too much of the details. Then there is a gain. There is a gain that there is the addition of the nucleotides. So I think this is an important concept. I will take that break. So we can have a 15-minute breaks, and if you don't mind, we are already behind the schedule, so it's like 15 minutes past 12. If we can come back at 12:30.
I have a question. So you mentioned about SNPs. For SNPs to be the predisposing factor for a particular cancer, is it necessary that they should be in the promoter region or the exotic region?
It can be anywhere, but the functionality has to be proved. So like basically, let's say if it is in the nearby the promoter, most of these SNPs are in the non-coding genes. That's the first part. Now if it is in the coding gene, it's a gold mine. Okay, so then we are basically done. You have everything in hand, but 99% of the cases they are in the non-coding genes. So I will discuss that part either in the 5' or 3' UTR region, or nearby the promoter, or nearby the enhancer region. So if it is in the nearby the promoter region, for example, then we need to find out by luciferase A or AMSA and other things that if you introduce that defect, then its activity is affected, that particular gene's activity is affected, and then of course then we have to do knock-in and knock-out studies. Only then we can say that. But at this time, from the statistics and like association studies or epidemiological studies, what mostly is happening is we say, okay, liver cancer is here, and there are 10. These are 10 SNPs that are present in Caucasian population in the tumor liver cancer, but not in the normal. Normal is like 5% of the. It may never be the case like 100% is present in the tumor and none is present in this one. So if that is the case, then you are really looking at something. So it is like the frequency is less. That's why there is the term. I think there is a statistical term by which it make a curve saying that LDX value, how much it goes in, and then you find that okay, this much shows the association as per in the tumor, but not in the this thing. So it kind of a correlative study. It's not a definitive study. That's why I gave a statement like if someone spends other 5 years in confirming that particular SNP, that's what it is. But it may be that SNP is not a real SNP. It is just a correlative. But these kind of studies are mostly done by epidemiologists, and they are statisticians. Okay, so again, like in my dear college, I'm not criticizing anyone. They have a very good role to play in all these things, but someone has to test these functionalities. And in fact, like at one point in time, NIH has some reservation about these association studies, but still like they sometimes like AMD, they are doing pretty good. Hemoglobin, they found out a SNP which is pretty good. So there are some good candidate SNPs are also there. What we are facing in here is a dilemma. Like if someone else has to, let's say you want to follow it, you cannot just blindly follow it. That is the candidate. You have to think about what you want to input, what you are going to get, whether your student is going to get a PhD or MSc or you will be stuck forever. Okay?